Hello everyone, my name is Simona Notova. I'm originally from Slovakia, but currently I'm living in Grenoble, France, where I'm doing my PhD. The city is located in the French Alps, and so do our institute CERMAL, where the main topic of research are carbohydrates. I am working under the supervision of Dr. Anne Amberti, and our group is focused on functional and structural study of lectins and glycosyl transferases. My PhD is a part of European training network called synBIOcarb. synBIOcarb brings together 15 early-stage researchers from all around the world and combines synthetic biology and glycobiology towards applications in diagnostics and targeted delivery. Glycobiology, the sweetest of biology, is focused on study of carbohydrates, or sugars if you prefer, and interactions with them. Are you imagining chocolate and candies? Sorry, I meant different kind of sugars. Do you know that every living cell is covered by the layer of sugar? This sweet shield, called glycocalyx, can protect the cell but also serve as a target for many viruses and bacteria, which by adhesion are able to enter the cell and cause the disease. The mechanism, how they reach the attachment, involves lectins, the proteins which interacts with sugars. Depending on lectin specificity, they can recognize different sugars and cells. What is interesting is that the type of sugars changes depending on physiological state of cells, which means that glycocalyx of healthy cells is different than, for example, of cancer cells. So what if we could use the lectins for detection of cancer? Great idea! Unfortunately, most of the natural occurring lectins has only single specificity, which means that they can bind to one type of sugar. And here comes the synthetic biology. We have designed and created proteins with new scaffold and double specificity. Why is it better? Aiming for two different sugars brings advantages like higher and more precise targeting of the cancer. The new proteins are called Janus lectins. The name is derived from Roman god Janus, who had as our proteins two faces. So what am I actually doing in my project? First, we decide what we want. Then, we design the gene and protein sequence. The gene is synthesized by the company. And after, the real lab work starts. First, we need to insert the gene into vector we want to, and like this we can transform the bacteria which produce our proteins. Protein expression in E. coli provides fast, cheap and easy production of Janus lectins with reasonable yields. In the next step, the protein is purified by series of chromatographies. The functionality of protein is tested by biophysical methods like surface plasmon resonance and isothermal titration calorimetry. And our biggest goal is to solve the structure of protein by SAX or X-ray crystallography. This involves protein crystallization, the process in which proteins form the crystals, fishing of crystals, and data collection at the synchrotron. Now, let's just hope that we are lucky and from the crystal diffraction we can see nice electron density map from which we can reconstruct 3D structures of proteins and ligands. This research can be adapted for the needs of other members of synBIOcarb network, and together we can use our growing knowledge in the analytical, diagnostical and therapeutical applications. This video and research could be realized thanks to financial support from European Union and Marie Sklodowska-Curie actions.